All right, I think we can get started. Today we're very excited to have Jason Yim from MIT, who is a third-year PhD in computer science, advised by Tommy Yakola and Regina. He previously has been a research engineer at DeepMind, and he also participated in multiple. He's the leading author of multiple very important work in the field of protein design, including RF diffusion that we mentioned multiple times. That also kind of is one of the reason that the work has been awarded the Nobel Prize. And so today, Jason's gonna teach us about how we can apply diffusion and flow matching on a wide range of key essential tasks in protein design, especially structure-based design. So without further ado, let's welcome Jason.
Okay, cool. So everyone can hear me, I presume. So yeah, feel free to ask questions. Maybe Gay, feel free to interrupt me or ask in the comments or unmute yourself. So the title of my talk today is Machine Learning Techniques for Biomolecular Generation. I've worked on this for my PhD, also for a little bit while at DeepMind, and I think for the past 3 years there's been a lot of progress. So it's kind of very cool to go over the work since then. So I'm gonna assume, like, just from basics that people don't have much knowledge about proteins, but I'll kind of go fast through some areas if there aren't any questions. So I think we kind of all know why proteins are important. They kind of evolved throughout life to do important functions like DNA replication, and but then there's a lot of things that we don't have good proteins for. And so in protein design, we need to engineer proteins for these specific needs. We've all, hopefully a lot of us went through the COVID pandemic, and so we know the need for vaccines and also drugs that protein design can help in. There's also been a lot of interesting work in like plastic degrading enzymes or enzymes for industrial applications as well. Oh, I hear some echo. Okay. And then another very cool application I think is genome editing, where we can engineer proteins that can do base pairing editing and like maybe make CRISPR more accessible or more efficient when we're trying to solve like genetic diseases. And when I think of protein modeling, I kind of think about it in like these stages. So this might be very basic to a lot of people, but just so we're all on the same page, DNA is this 4-letter vocabulary. So you can have like the 4 nucleotides. And in sequence, when we think about a protein sequence, each kind of residue in a protein can take one of 20 letters, and each of these letters corresponds to an amino acid type. Yeah, so in like central dogma, you go from like DNA sequence, DNA, RNA, protein. So here, like DNA gets transcribed into a protein sequence, and then that sequence folds up in water to become a 3D structure, and that's where the 3D coordinates come. And then we have this view of the 3D structure as a static object, but then if you actually kind of look in the body, proteins are actually dynamic. They're kind of binding. There's reactions happening, and that's kind of what we associate as function, as when the 3D structures interact with each other. That's kind of the hierarchy of how you go from DNA to the protein function. And I think as we've known, kind of like why I guess teaching this class is that machine learning has revolutionized kind of protein modeling, not just protein design in my slide title here. I think we all know AlphaFold 2 at this point. This is the task of taking a protein sequence and trying to determine the 3D structure out of that. This is AlphaFold 2. There's also been AlphaFold 1 and 3. And then there's also been this inverse task where, given the structure, you want to get back to the sequence that could fold into it. And so this is another paper called Protein PNN that was by the David Baker lab that works extremely well. And then what more recently has come about is trying to go from a function to a structure. And so this is what I was an author on and was also published in Nature, that RF diffusion that I'll be talking about today. And I just also wanted to give this timeline of how like prevalent and how popular generative models in proteins have become. So there was like a point, I think up until like 2022, where AlphaFold 2 and like diffusion models just kind of were published and kind of getting popular at the time. And then after that, there was just this huge explosion of diffusion models just to generate new protein structures. And so we have a review that you can look at that kind of outlines different approaches that people have taken. And I'm quite fortunate to have been part of several of these papers. But I think it's great to see there's a emerging community of a lot of people that want to investigate how to do generative modeling better for proteins, but also just in generally in biology. So I think that's why this is um, like a very rapidly growing field that's really good to get into research if you're aiming to do a PhD or already are in your PhD. Um, some might call it competitive, but I think, it's kind of competitive for a reason, which is why it's very impactful. It's won a Nobel Prize and stuff. So it's something good to get into if you're very interested in biology and machine learning. So kind of when we talk about protein design, kind of how do we fit generative models into this pipeline? So the first task I think of in protein design is one, the backbone generation to generate kind of the skeleton of the protein structure. And this might kind of seem counter to it, but we'll see kind of why this gives you a little bit more freedom later on. And then kind of once you generate the structure, you're going to generate the sequence that can fold into that structure, most likely. And then once you generate both the backbone and the sequence, you can do something called virtual screening, where you'll generate a lot of proteins and you'll pick out which one is good and which ones are bad. And then once you kind of pick out the ones that are good, you're going to run them into the wet lab and actually experimentally validate them. So this flask is meant to kind of actually see if the proteins can express, if they bind, have some function in them. There's a lot of different ways to experimentally validate your protein. And so today's lecture is going to be focused on these first two. And for virtual screening, we typically use like the folding methods to tell like if a protein does exist or not. I'll go a little bit into that. And then this experimental validation that is kind of beyond the scope of today's lecture. Oh, I guess I will talk about RF diffusion and experimental validation we have done, but kind of like more complicated experimental validation, I won't go into details. So let's just take a step back and like, what is the goal of AI and biomolecular design? Once again, like how I view it is we want to combine generation optimization into one pipeline. So what we talk about is generation, where you can use generative AI to create these novel and highly diverse molecular libraries that can then be fed into the wet lab experiments. And the ultimate goal, I think, is that if you can actually close this loop. So once you run your experiments, you can get this data back if it works or not, like failure success. And then you can feed that back into generative AI and have this system of how it continuously improves itself. And that is one of the major goals of this that is very hard for humans to do, that AI can excel at. When data is like very high dimensional, we don't have a lot of it. AI can help us design new proteins much faster than previously. Yeah, so today I'll be going over first protein backbone generation methods. These are two papers that I've written, and then we'll talk about how this has been used for real protein design tasks with RF diffusion. And then I'll talk a little bit more about very recent things of how now we're trying to combine sequence and structure generation together. And this is going to, I believe, lead to new ways to view protein design that will come soon. And then just a very brief view over like the outlook. Okay, so I think hopefully most people here know what diffusion models are. My favorite instance is OpenAI's Sora model, where you give it this prompt, like in text form, and then the model is going to generate you a video or an image or audio that matches the prompt really well. And so this was, to the layman, to the general audience, this was kind of one of the wow moments of where generative models really took off. And to go through like a very high level of how diffusion models work, training and generation kind of works as follows. This is going to be at a high level, where you first take an image, you add some noise to it, and here is just Gaussian noise, and then you train the neural network, which is the model here, to denoise it, to get back to the original image. And if you can do this task really well for different amounts of noise, then you can start from pure noise, and then you can take these iterative steps where at the very end, the model is going to denoise enough of it that you end up with the real like a real image. And this has led to a lot of breakthroughs in diffusion models. So kind of how do we translate to protein backbones? Well, first, kind of what do we want out of diffusion for protein backbones? One is we want to make sure we generate high quality structures. And then two, make sure that they're diverse. So we don't just generate the same thing over and over again. But also, I think an important point is to generate novel structures. We already know a lot about proteins and have a lot of protein structures already, but we just reusing them we don't need AI for that. We actually need to generate brand new structures and brand new proteins so that they can solve these new tasks. Otherwise, there's kind of like not a point to this exercise that we're doing. And then the last thing I think that's kind of often forgotten, but is very important, is that you also want to have this guiding star of what is the function that I want to accomplish, whether it's like designing a binder or like an enzyme, and then more down the line of making a new drug or a vaccine and stuff. Those are all important considerations that we want to be considering. Okay, so to start off, before you think about like general modeling, you have to think about like how do I want to even represent a protein structure? And this is a not so trivial question that there's a direct answer to. There's this one option where if you model every protein as like a 3D coordinate, then you can just do like Gaussian diffusion and let them move around and then try to denoise that. And so this lets you have good control over where the atoms go, but then the bonds are like breaking and stuff. And so you're kind of losing the physical intuition of like what makes up a protein. And actually, there's a there's a paper back in 2022, which is one of the very first papers in diffusion models that I was one of the first co-authors on, where we kind of tried this out with the C alpha carbons of a protein. And it kind of worked. Like this here's like one generation sample of the diffusion model that it could like generate these proteins. But then we found this was kind of like difficult to scale, and it wasn't also giving like great performance at the time. So that was then, and I was at MIT where I was running my jobs on like 2 GPUs. And then lately we've seen people that shows that you can actually scale this up and start getting very good results. So this option one, we kind of abandoned it back then, but it kind of also shows that deep learning with enough scale, you can actually get rid of the problems that arise when you're modeling every 3D coordinate. The other option that you can think about is to model only the torsion angles that make up the backbone. So if you look at a protein structure along the backbone, you can see that they always share these 4 atoms between every single residue. And between them, there's these canonical angles, dihedral angles, that you can modulate to of how the protein unfolds. And this is nice because you're following the intuition, the physical intuition of proteins more properly, that the bonds should never break. But then it's really hard to control where you're placing the atoms. So here it's something called the lever arm effect, where given a protein, if you turn this angle a little bit, then it's going to result in a major change into the protein structure. And so when you have something that's stochastic like diffusion, this is quite difficult to control. And even in non-diffusion models, it was also difficult to control and really hard to optimize. So you had like less control. And then so there's a third option, which back then we decided to go with, was to model with these frames along the chain. And so I'll go more into details of what this means exactly, but you can imagine this is something in between option 1 and 2. You have this precise control over where the atoms go, but you can also use physical intuition and keep some of the bonds fixed throughout. And so how do you construct these frames is that let's look at the protein backbone atoms. And again, here's the canonical atoms along each residue. And what we can do is take the C alpha in the center, use that as like the translation in R3, and then build out a rotation from the vectors that go from C alpha to the nitrogen and C alpha to the other carbon. And then you can do something called Gram-Schmidt to end up with this third vector, which is just the orthogonal vector of the cross product, and construct a rotation out of it. And so this is nice because these 3 and even these 4 atoms are always going to be present no matter like what the residue types are. And so you can always construct this frame for every residue without knowing what the sequence or the residue identity is. And that's quite crucial because you're not worrying about what the sequence is now. You're just trying to model the structure by using this canonical representation. And so kind of once you construct a protein like this, how you would sample from it is that you would kind of start with noise, and then you kind of go noise, then go back to the structure and also force. So this is defined by 2 processes: the form process where you're noising the structure towards pure noise, and then there's also the reverse process where you're sampling. You're starting from pure noise and going back to the structure. So hopefully these 2 equations are familiar. I think Gad told me that the audience here knows something about diffusion models, so hopefully you've seen these equations. I'll go more into detail about how exactly these are instantiated for protein backbones. But the main thing to note here is that we're trying to learn this object, which is the score function, because we don't know what the score function is analytically, and so this has to be learned by a neural network. Okay, so frame diffusion, how do you diffuse a frame? So as I mentioned, the frame consists of two parts, which is this translation, the C alpha at the center, and then the rotation matrix that makes up, the other atoms in the backbones. And so we need to diffuse both objects independently of each other. So diffusion for translations happens in R3, where this is over the rotation manifold, which is called SO3. And here's just a quick intuition of how it looks, is that you can clearly see the translations are moving, but then also the frames are rotating as well as you add more and more noise until it all becomes jumbled up and you can't tell. I still keep the lines here to show kind of the chain so that you know which residue is next to each one. And so kind of just a little bit of math. In R3, you're just adding Brownian motion, so sampling from a normal distribution. Whereas for rotations, you have to do a little bit of work to apply a normal distribution or a Brownian motion onto this SO3. Because SO3 is compact, as in if you rotate 360 degrees, you're going to end up at the same place. You have to kind of be careful about the boundary conditions. So I'm not going to go into details of kind of exactly what the distribution looks like, but it's something called the isotropic Gaussian on SO3. It has this kind of a functional form based off exponential mappings and stuff. But the thing is we can actually sample from it, and it works in this diffusion setup. So kind of as you see at time 0, we have the data. So here is just one rotation. But then as time goes towards 1 or capital T, you can see there's more variance. The variance increases, and the samples becomes more noisy. So it becomes more of a stochastic distribution rather than just something centered at the data. Yeah, and so frame diffusion, now that we've gone over kind of how the diffusion more or less works, training generation kind of works very similarly to how you would do it for images. So first, you would take a protein structure out of the PDB, and then you would construct the frames out of it. So this frame corresponds to this protein structure in 3D. Oh, oops. And then you would add noise, as we just discussed, and you train the neural network to uncorrupt it, to get back to what the original structure was. And if you can do this very successfully and really well, then you can start from pure noise, and then you can use neural networks to sample new protein structures. Now, in terms of the model architecture of how you actually learn this function, there's, I think everyone here has heard of transformers and how good attention is. And so AlphaFold 2, one of the major innovations there was to use attention throughout the whole architecture. And so it's something that we adopted here. We didn't use the AlphaFold 2 architecture exactly because it is quite expensive, and in my belief, it was kind of overkill for this task. So we kind of took ideas from AlphaFold 2 and how we constructed this model. I think AlphaFold 2, there's two main ideas. One is this invariant point attention, which is that residues that are closer to each other attend more to them. So here, this frame, I kind of drew this out on an iPad, so it's not super polished, but let's say this frame here is going to attend more to frames that are close by to it geometrically. Frames that are farther away are not going to have a lot of attention on them. But the thing is you still want to know about them. And so that's what this transformer also does, which allows global interactions for the model to communicate with residues and frames that are very far away. And so you want to have both of these into a single neural network. I'm not going to draw the wiring diagrams because it's going to be too much to understand in a lecture. But yeah, in the neural network, we always have these node features, which also includes the time embedding and also the positional encodings, and then also these edge features, which kind of captures how far two residues are from each other, both geometrically and relative to their positional indices. And of course, you have the frames that, as I discussed, those are also going to be talking to each other based off the invariant point attention. And so in the neural network, we do both the positional attention and the spatial attention multiple times. And this L stands for the layers, so you do this multiple times so that the model builds up a very rich understanding of the current protein structure. And what we found is that, yeah, having both attentions is very important for this model to kind of work properly. The other thing to mention is invariances and equivariances. So intuitively, like if you rotate a protein, then it's still gonna be the same protein basically. So we want a way to sample proteins that if you rotate it, the noise, then it's also gonna rotate the structure. So for instance, let's say this is a noise I sample, and then if I run the model, it's gonna sample this protein. But if I rotate this noise, then the sampled protein should also be rotated. This is desirable because if you don't do this, then the model is going to have to learn a much more complicated neural network function that is going to have to map every single rotation of the noise to the same protein, and that is not very efficient to do for a neural network. So this requires two things. One is you need to have this equivariant score model, and this architecture lets you have SE3 equivariance in there. And then you also have to have translation invariance, which is just keeping the noise and every single step of the process always zero center to make it like translation invariant. Yeah, and so like when I mentioned the score being invariant or equivariant, that's just what this object is, which is learned by a neural network. That has to be equivariant to sample equivariant proteins. Yeah, so then how do the actual samples look like? So the first task you think about in general models is just how well do you learn the data distribution? And so this is what we refer to as unconditioned generation, where you just give the model noise as input and no other conditioning, and just say, generate me a protein, it can be any protein. And so here's 8 different noise samples, and then you give it to the model, and you run diffusion, and it's going to sample realistic proteins out of all of them. And the cool thing is, like, yeah, all of these are different proteins, like they're highly diverse in there. So that's kind of one thing that we've accomplished. But then, yeah, so like that's how you generate protein, but how do you actually evaluate how well you're doing? The main metric that we use for protein generative models is something called designability, where we test if a sequence could exist that could fold into the backbone that we designed. So to kind of go this step by step, you first generate the backbone, and then you're going to use another model, PROMQ-BNN, if you remember at the very beginning. Sorry? I think there's a question?
No, I think someone accidentally unmuted.
Oh, okay. Okay, yeah, feel free to interrupt me. Yeah, so from the backbone, you can use something called PROTPNN to generate the sequence. And what we do is we actually generate 8 sequences because remember, everything is probabilistic here. And so there's always going to be this distribution over sequences and structures. And so it's kind of advised to sample multiple sequences that can generate into the backbone. And then we're going to use a folding network. Here we use ESM fold. You can always also use AlphaFold to generate a prediction for every single sequence. And then if you do this, you can take every one of these predictions and test how well it matches your original backbone. So it's kind of like this information bottleneck that you want to see if ESM fold or AlphaFold can predict what your original structure was. And the intuition behind this is that you're trying to find a sequence that ESM fold or AlphaFold will like, that will that could try to recapitulate what your original backbone was. And so we call something called designable. Designable meaning that there's a sequence if any of these predictions has an RMSD less than 2 angstroms to the original backbone. So this has worked quite well. People have found that this correlates well with wet labs as binders, or just telling if a designed AI designed protein would actually become a real protein. But that's just not all. We also, as I said, want to have diversity. So what we do is that after we design a bunch of proteins, we structurally cluster them. And so there's a lot of algorithms for this where you can take 2 proteins and construct a score between 0 and 1 of how similar they are. You can construct this matrix of like every pairwise TM score of how similar they are, and then use a clustering algorithm to determine like how many unique proteins you have based off this graph. So then what we do for diversity is we typically report the number or like fractions of clusters, and this tells you how diverse your generative model is. And the last thing is something called novelty, where we see how similar these backbones, if they're designable, to any known structure in PDB or AFDB. And this is, subjectively maybe one of the most interesting things to see if your model can actually generalize outside of its distribution. So for frame diff, that's what this diffusion model is called in the paper. We took these just single chain backbone structures from PDB. We kept it to between 60 and 512 residues. We also did a little bit more filtering, and in the total, we ended up with like about 20,000 structures. And I see this is like a lower limit. You can definitely loosen your filters so you have more structures, but what we're going after here was like a very high quality set of proteins that we could train on. And what we found is like, so this is kind of the designability. This is this RMSD here of like how close the prediction in the backbone is. And like I said, you want this to be below this line of 2 RMSD. And so we can see that across lengths, the model does quite well at length 100. Like most of them are designable, but then as the protein gets bigger and bigger, it becomes a lot harder to satisfy this RMSD threshold. And this SATM score is another way to measure kind of how well your designable protein is, but it's not used anymore. So yeah, we see length is one factor, that the bigger your protein gets, the harder it is to generate a high quality protein, which I think intuitively makes sense. The bigger the protein, the harder it is to generate a good one. But we also see that the protein has very very secondary structure in it. So for those that are like know something about bio and protein structures, you can measure how many alpha helices there are and how many beta sheets there are. And you can see that our model isn't just collapsing onto like one mode, but samples a diverse range of secondary structures. So there's also this diversity in terms of like these structural motifs. And we also see that the model can go beyond the PDB. So again, this RMSD like is how designable it is. You want this to be below 2. And this is the similarity to the training set. So if you're up here, 1, that means you're basically sampling something that's in your training sets. So we want this to be as low as possible, and we want this RMS to be as low as possible. And so you see most of them are here, as in it's basically sampling a lot of things from the training set, but there are things that are very different from the training set. So I've taken 2 here, one this is in the red box and one in the green box, and I chose these because they're also very different lengths. So this is kind of like a medium-sized protein of length 135, and ESMFold really likes it. The RMSD is below 1. Its confidence, this PLDDT, is like 80 or above. And then the closest protein in the PDB to this, roughly it looks the same, but like it is functionally very different. The TM score is 0.47, which means it's very different. And there's this bigger protein where the closest protein in the PDB is like very extremely different from it. So this is to show that these generative models don't just memorize the training set, but they also go beyond it. Okay, so that was frame diff. This was back in like 2022, so like not too long ago, but there's been so much more happening since then. And one thing that's happened, as you guys heard, is flow matching, where diffusion models have kind of switched to flows. And the general motivation behind it is that they're functionally very similar, but flows are kind of simpler with diffusion, and you get the equal performance out of them. The main thing is that you don't have to deal with like stochastic differential equations, although you definitely can, but you can instead just deal with these interpolations between data and noise, which is just governed by this very simple function here that everyone should be familiar with. And so it's much easier to think about flows and reason over them. And one of the benefits of that is that you get more exploration options to explore over different hyperparameters. And one of the most important things that we found is the noise schedule, like how fast you inject noise and how fast you remove noise. And so here's kind of a illustration. So on the left is frame diff, the model I just went over. And here's frame flow, which is the model with flow matching applied. So you can tell that the one on the left is like more noisy, is like going over the place. And then frame flow is like much more smooth and going to like its final destination. As a result of this, another benefit you get is that you can sample a lot faster because the model doesn't have to resolve so much noise in the sampling process. You can take a lot fewer steps and sample a lot faster when you're doing flow matching, which is not that big of an issue for proteins because the models are like not gigantic yet. But for other domains such as images or language, you really do care about efficiency of your model. But I think protein models are also getting very big, so efficiency is also becoming at the top of a lot of people's minds. The other thing that's important is like we previously mentioned, like, yeah, you definitely need this like spatial attention mechanism called IPA, but like what do you scale though? If you want to make the model bigger, do you want to add more parameters to the spatial attention, or do you want to add more parameters to the positional attention, which is just a standard transformer? And so we did a lot of architecture exploration and found that making the transformer bigger leads to better performance. So this is not too surprising to people in other domains like images and language, where the transformer, it's a very good scaling architecture where if you add more parameters, then it typically does better. So this kind of that was like surprising to people who work on proteins who want to stay more physical as possible. But numbers speak louder than words, so scaling transformer just performed better in this context. And so then function generation, here we sampled 100 lengths from 70, 200, 300, 200, 300 lengths. And then these are the three metrics that I previously described. You can see that frame diff gets much lower performance than frame flow. So this flow matching switch plus some architecture switch led to way better improvements in terms of across all three metrics. And yeah, we also end up with like very novel structures that look very cool. So if there's like a histogram of of proteins and like how similar they are to known protein structures, we can see that these are very quite different than things in the past, like more different than frame diff. Right. So this is to generate a protein structure of how well you learn them, but we also want to generate functional structures. So a lot of protein functions can be defined through like a motif. It's just a few residues that are responsible for the majority of a protein's function. So like antibodies will bind based off like their motif to viruses based off that. And the binding happens not because of most of the protein, but just because of a few proteins that make up the interaction highlighted in purple here. And so motif scaffolding, if you can identify what the functional part of your protein is, you can then kind of reimagine what the rest of the protein can look like. So here's like a real protein in nature. We're going to take this blue part and then generate a new protein that keeps this while generating the rest of it. And you can think of this as like an image painting task, where in an image, you're like mask half the image out and the model has to generate the other half of the image. But you can see that like the beginning and the end are very different. This is like a double-decker bus. This is like a single-decker bus. And so it's like a similar thing for proteins. We're like going to cut half the protein and we're going to ask the model to generate the other half, but hopefully it's like a different protein than it was previously. So how do you do this training is very simple, but that's kind of the beauty of this, where you take a protein structure, you're going to randomly select part of the protein as the motif, and then you're going to noise the scaffold, so like add random noise to it, and the model is going to have to like try to denoise it or predict what the original structure was. And then so if you do that, then you can take the same motif, and the model is going to generate very different scaffolds for each of them. And then we test this with this benchmark that was previously shown in RF diffusion across like a lot of different bio functions. So like from epitope presentations to active sites, interactions. And so this is just like a showcase. I'm not going to go into any details on any of these, that we were able to successfully scaffold all of these cases that are known to be very important in therapeutics or industrial applications. Yeah, so FrameFlow I think one of the benefits of it is that it gets this diversity of scaffolds much more than previously done. So like just to highlight it again, I give it the same motif in all of these cases. These are motifs taken from these PDB IDs, and the model is able to generate like very different scaffolds. So here, some of them have like a lot of beta sheets, or some have like just pure alpha helices. And so you get this very diversity of scaffolds. So just like a takeaway of this section is that in diffusion models for protein design, we kind of wanted these four things: high quality, diverse, novel, and functional structures. And we've kind of done a lot of work over the past few years to accomplish these. But then kind of everything I was explaining up until now has just always been on a computer. You don't believe me that these are real proteins unless we test them out in the real lab. And so we actually want to apply these to real protein design tasks. And so that's kind of what led to RF diffusion, where we've been collaborating a lot with the David Baker group at the Institute of Protein Design, and did this like huge collaboration to do RF diffusion together. So that's what I'm going to talk about now. But I guess I'll take a moment if there's any questions.
Yeah, there's one.
Yep.
Hi. So, can you, in the scenario of biomolecule generation, can you maybe say some comments like the advantage of flow matching over diffusion and vice versa?
Can you repeat that again? Couldn't hear it well.
So I think he's asking about the like concrete advantage of flow matching over diffusion on biomedical applications.
Yeah, and I also want to know the reverse way, like the advantage of diffusion over flow matching.
Oh, and whether there might be an advantage for diffusion as well.
Yeah. So look, the way I view it is that flows and diffusions are two views of the same thing. one, like they're all part of this way of iteratively improving your samples and iteratively sampling something. Flows gives you one perspective that's based off like linear equations, and then diffusions are based off a stochastic differential equation. Diffusions are nice because they're very closely related to like probability theory. Like SDEs, you can write down the probability that you're trying to sample. Whereas flows, they're very nice because they're very close to optimization problems and like control theory problems. And so often, like what we do now is that we go back and forth between the two. Like I'll have a flow model, but then at any point I could maybe want to switch it to a diffusion model. But then there are certain cases where one is better than the other. So in flows, like I mentioned, one of the best things is hyperparameter exploration. And so like Stable Diffusion 3, for instance, they showed explicitly that flows can improve over diffusion because you can sweep over more hyperparameters there. And that's something that we find in this paper as well, that we can sweep over the noise schedule a lot easier. Now, there are some other papers that say diffusion is better because like adding noise in there kind of helps. So it's kind of like an open question of like which is better in which case. At least for protein structures, we're finding like flows. If you want to do like Riemannian stuff on frames, flows is better because it's much easier to define flows on manifolds. So like long story short, the answer is like it depends for proteins. Currently, we're finding flows are better because you can do better hyperparameter explorations, but that might change next week or in 2 weeks. It's always going back and forth. The thing is, like, they're all based off the same concepts, the same principles.
Another question is, I've heard from some friends that diffusion may produce higher quality image compared with Fibonacci. I want to clarify that, like for protein generation or molecular generation, so improving the quality or improving the efficiency, which one has a larger optimization space, like
Yeah, yeah. Yeah, so for flows, I would say there's bigger improvements for efficiency because it's less like, yeah, it's like less noisy. So you can use a lot of ODE integration methods. Diffusion, I would say, is kind of better if you want to do more complicated like Bayesian or statistical stuff. Because yeah, it's very easy to like write down a probability distribution of diffusion and do kind of like more conditioning mechanisms with it. But like again, you could maybe think about, okay, if I want to do efficiency, then maybe I'll think about flows, and maybe if I want to do more complicated conditioning tasks or like Bayesian inference tasks, then diffusion might be better. So yeah. And then some people have different views than what I do, so it varies what people say, like what one is better for than the other.
The real question I want to ask us is, like, which one is more, like, more challenging to put into real use of those generic models? Like, the quality is undesirable or the efficiency is more concerned?
I think diffusion is harder to put into a model. It's like it, yeah, it requires more debugging and yeah, flows are nice because, like I said, they're quite simple. So I would start off with flows, and then if you're doing research, start off with flows, then try diffusion, and then do your own exploration because I think it's going to vary between your application, like what's better.
I guess one question I have is, do you have any intuitive explanation of why flow matching has higher diversity? Because we thought that if you have noise, then you probably have more diversity.
Yeah, I think because flows are better on rotation spaces in this case. So on Riemannian manifolds, flows are quite elegant and simple. Whereas for diffusion, although you kind of know this IGS3 diffusion kernel, there are some like approximations you have to do in calculating the score of a diffusion model on manifolds. So by that, like flows don't require approximation, diffusions kind of require approximation. So in my experience, I think flows are a little bit more principled, and that might be why it's leading to a better distribution.
Any more questions? And like audience on Zoom, do you have questions? Cool. I think we can move ahead then.
Cool. Okay. All right, so Rosetta Fold diffusion was a combination of two ideas, basically. So at the IPD and David Baker's lab, they've always been, I guess, advocating for using pre-trained models. So we have a model called Rosetta Fold 2 that's very similar to AlphaFold 2, and it's like a pre-trained protein structure prediction network. And then we came in with diffusion, and then the answer was like, okay, let's combine Rosetta Fold 2 and diffusion in some way. So like we use the strength of a pre-trained model, but also use the algorithmic advantages of diffusion to do protein design. And so there's like maybe like three main things on R diffusion that kind of made it work. So if you look at the Rosetta Fold architecture, this is taken off, I think they're figure 1. It usually would take in a multiple sequence alignment and like 2D templates, as well as like the 3D structures in terms of a coordinates. But it's like it's trained this way, but you can hack the model so that you only use its output frames and the template coordinates and feed that back into each other. And so although Rosetta Fold was trained end-to-end with all these extra features, you can fine-tune it for diffusion because you have access or places to put in the frames and train it in a diffusion way. The second thing is like this transfer learning, which is that like structure prediction and diffusion kind of share similar objectives. So in structure prediction, you typically use something called the fate loss. And here you're looking at like how well the frames align per residue. And then in diffusion loss, you're kind of trying to match the frames to each other. And so what you can show that is that these losses, they share the same minimizer, but they have different gradients during training. But because of this, because of the similarities, you can pre-train on this task and then use SC3 diffusion to fine-tune it and turn it into a diffusion module. So like this is how RF diffusion was trained. You would first pre-train with the fate loss, and then you would use this like SC3 diffusion loss. In the paper, we have slight modifications in the loss, but not worth going into right now. And this will lead to something called the RF diffusion base model. And so then we can do the same unconditional generation with RF diffusion that we did for frame diff. And you can see that with the pre-training, it kind of generates much larger proteins, and they're like very diverse that the pre-training kind of gave it. And so this ended up becoming like quite a bit better than frame diff at the time. And then we can also quantify novelty again. And we can see that this actually gives a much more novel distribution of proteins, even though it was pre-trained. So like this is the similarity in proteins, and like a lot of them are below 0.5. And so there's like a density plot. And so across these 4 lengths, you can see, yeah, the model generates very novel proteins. Here's one AI generated protein is the most similar to it. And you can see the only thing that's similar is this like beta plated sheet that matches, but otherwise it's like very, very different. But then now we also wanted to conditional training. So we took this base model and then we fine-tuned it for like enzymes, protein-protein interactions, and something called potential guidance. And so this is why I think R diffusion is so widely used, is because we adapted it for a lot of different tasks. So apart from the conditional scheme, you can also add symmetric noise, do binary design, full generation motif scaffolding, and also combine things for symmetric scaffolding. So for binary generation, like here's just like an illustration, is that you give the model the target you want to bind to, and you can even specify the epitopes, and you can generate the binder that'll kind of bind tightly at that spot. You can do binary design, but as a motif scaffolding way, where let's say like at the top of this crown virus that kind of these motifs will bind to it. So you kind of want to generate the rest of the protein, but also have it be symmetric in there as it generates the binder. So binary design, it's quite a simple idea, which is while you train the model, you kind of tell the model where to bind to. So you call these hotspot residues. So like here's 2 sites. You can tell the model to bind me here or bind me there, and the model is going to be able to like follow these guiding hotspots and generate binders at those. And so we find it to be important to kind of guide where, because if you don't specify where to bind, the model might always bind to the same spot rather than the 2 spots that we know it should bind to. The other thing is that you can also condition on the fold topology so that you can bind there, but you can also control what kind of protein you're doing. So here's like a ferredoxin fold, and you can also just have it be a pure helical bundle. Or you can tell the model to come with its own fold and generate, conditioned on the hotspot residues here. And then with the Baker lab, it was crucial to also test these out in the wet lab. So here's like 4 proteins where we're also able to test like the binding affinity, which is this KD metric. And like basically like the faster it drops off is the tighter the binding is. You see that some of them are very tight and some of them are like kind of so tight, but there's like a varying wet lab success rate of these AI generated binders. And then the main thing is that compared to previous approaches, like our Rosetta design was one of the most popular approaches for binder design. This is like a physics-based method. This like deep learning AI-based method was orders of magnitude way higher. So for some of these cases, I think the success was like 0.1%, and this our diffusion was just able to like knock it out of the park and get orders of magnitude higher success rate. And then they were also able to go and actually characterize the structures, like solve the structure of the binding site. And then so kind of, and the design is in like purple or blue, and then the real structure is in gray here. And you can see that it's actually a very close connection of like what we designed, what we wanted. Yeah, so I'm gonna go a little bit faster because I'm running out of time, but this is symmetric protein design where you apply diffusion symmetrically to generate the symmetric complex. And then we're also able to test this out and verify that the proteins actually became these symmetric complexes. We also combine scaffolding and symmetry to generate symmetric scaffolds. And of course, these are also tested out, and we found that they are actually active. And there's also been like follow-ups where now we're doing like all atom diffusion, and there's gonna be a follow-up after this where there's also like flow matching, but that's gonna come out I think sometime later this year. And then another big thing is the antibody design, where we fine-tuned our diffusion to generate specific antibodies for like the different target classes there. Okay, yeah. So then the last thing I want to end off with is co-design, where we've always thought about doing structure first, then sequence. And this is kind of nice because by breaking down the task, you could focus on just the structure by itself. And that led to, a lot of cool results in RF diffusion. But then, like, what if you try to do both together? You can imagine if I do this jointly, then maybe the model can be smarter about reasoning about both structure and the sequence at the same time. And so this task is something we call co-design. And so to do structure and sequence modeling, as I discussed, structure generation with like diffusion or flows is fine. We have a tool for that, but then sequence generation is not so easy to do. In particular, we want these to be compatible. We know language models and sequence generation works there, but language models typically go from left to right, and it's also like a discrete process where you unveil one at a time. Instead, we don't know the order of how you should generate the protein. And so that way we have to learn any order of generation. That's its flexibility. Yeah, because we don't know like what order is optimal for proteins. And then we also want it to be in continuous time to be compatible with the SC3 flows. So kind of like what we developed is something called discrete flows. Other people call it discrete flow matching. I should probably update my slides called discrete flow matching. But the general idea is like as t goes from 0, which everything is x or masked, it's going to slowly unveil parts of the sequence or here characters until it ends up becoming like a real sentence at the very end. And the nice thing here is like, yeah, t tells you how much or what proportion of your sequence is going to be masked. And then at 1, everything is going to be unmasked there. And so one thing you notice here is that once a mask goes to unmask, it stays that way. But with discrete flows, a very cool thing is that you can introduce randomness and like teach the model to also correct itself. So here, a model, if it flips to a character, it might also have like the probability to flip back because it might be like, okay, I screwed up later on. And so it can also correct itself. And that we found is very important. And other people have found it to be very important as well when they're doing discrete flow matching over a language. So the whole theory of this is based on something called continuous time markup chains, which is a really beautiful, elegant resultant probability and stochastic processes, really fun to learn about. But here's like a very quick overview of how it works. Let's say kind of at the very beginning of my diffusion or flow, my state is going to be at A here. And then as time goes, there's going to be a time here where I'm going to make this jump. Yep, sorry. Is there a question? I don't think so. Okay. Yeah, so there's going to be like first jump. And so, yeah, it's going to become B here. And then like randomly, there's going to be this other probability, and the model might decide to jump here as well. So this is like going to be stochastic. So these times are random, but it's going to be weighted by kind of what this rate matrix is, how fast you should jump to each other. So here, like, yeah, for instance, it jumped back to this blue dot, but then like the final jump here before it ends might jump finally to this a red dot, and this time goes from like 0 to 1. And so the thing that you need here to learn is this rate matrix RT, which is the same thing as like the velocity field in flow matching. And so the nice thing about discrete flow matching is that you have the same objective as in really popular language models. And so, yeah, I guess this is not going to be easy to explain so quickly, but I think the main thing to pay attention to is that to define this rate matrix, you need to learn this denoising probability, which is the probability of the final state given your current state. And this is just trained with cross entropy, which is the same as autoregressive models that are used to train GPT-2, 3, 4. It's the same objective, but now you can use that to do discrete flows. And then like how to define a conditional rate matrix. If you want to like into the math of it, you have this Kolmogorov equation, which basically says like the influx and outflux of your probability mass has to stay constant. Like how your probability changes, you have to kind of make sure it's either equal to each other or kind of going in the right direction. And so to solve this rate matrix, you have to solve this Kolmogorov equation for whatever process you have. So what we typically use is either a uniform, like starting from random characters and going to a data character, or starting from like a masked character and ending up at like a real sequence. And so the one that's most popular is using this masked rate matrix. Sorry, I'm just going a little fast because I'm running out of time. And the other thing is, like, again, I mentioned the stochasticity that we found to be quite important. You can also like control how much stochasticity or how much the model kind of goes back and forth between like a mask and the real token. So here for the very high stochasticity, you can see the model is kind of jumping a ton there. So let's put like one more time. Like this is jumping very rapidly, whereas this one is like not jumping that much. And this is kind of like a hyperparameter you're going to have to tune based off what your task is. Yeah, and so like how do you add stochasticity? Basically define a detailed balance equation of like how do I add more stochasticity without changing the probability mass at a certain time. And so you can define a stochastic rate matrix where this RTDB is like this random quantity. And something I think I really like about this is like, at least in flows, it's very easy to reason about continuous and discrete because the equations look very similar to each other. So in continuous case, you also have a Fokker-Planck equation of like how the probability distribution evolves. You can also define that for the discrete case, except here you have a velocity field VT, and here you have a rate matrix RT. And I'm not going to go over the rest of the equations, but it's very interesting to think about that whatever is in continuous space, there's also a same concept in discrete space that lets you reason about it. And so I think for people who are, maybe more interested in like theoretical machine learning or developing a lot of machine learning methods, there's a lot of work in how to do like continuous and discrete flow matching together jointly. And I think it's an exciting direction that you can look into as like a PhD topic. And so kind of what we do in this paper is something called multi-flow, where you're flowing over the structure space, which is continuous, but you're also flowing in sequence space, which is, this like AT variable. And so then if you like combine it together, then we are able to do sequence and structure co-design where both the sequence and the structure are being generated at the same time rather than two steps. And the cool thing about this that I really like is that we're unlocking a single model to do every single task in protein like modeling for sequence and structure. So for instance, like let's say I have like a noisy, or like pure noise for the sequence and mass structure. Code generation is sampling things together at the same time, but then you can also do it piecewise. You can maybe first do the sequence and do forward folding to generate the structure conditioned on the sequence, or you can do the structure first and then do inverse folding to generate the sequence conditioned on the structure. And multi-flow allows you to do every combination of this, as I'll explain. So there's two time variables with multi-flow. One is the time for how fast the structure is being denoised, and then this like t tilde, which is the time for how the sequence comes from mass to a real sequence. And so then in the model, we actually train on every combination of the two, so the model kind of knows how to deal with each situation. And a neural network is the same as the one in different frame flow, but instead we have like a sequence prediction at the very end. And so then like this matrix that I showed previously now corresponds to different time variables, where for different combinations of T, you can simulate different like tasks or generation tasks in your protein model. And so then like this task of doing both co-design of generating sequence and structure, we evaluated like, yeah, this comes out of our model, and then we do the prediction and look at the success. We can look at the designability, diversity and novelty that I previously explained. And compared to previous models that were attempting to do co-design, we show that we're like, two to 3 times better than the next best model in terms of designability, and way better in terms of the other metrics. Now we can also see kind of like compared to co-design, what if you did the two-stage stuff where you use protein PNN to generate a sequence on the backbone? And here we see that using multiflow for protein PNN1, that it is like very similar. So by doing things jointly, we get almost on par performance with doing it in a two-stage way. So this can allow you to be more efficient and let you reason more about sequence and structure together, do cool things with it. And then for folding results, here we actually like held out PTB structures, and then we use multiflow to generate the sequence conditioned on the structure. And we measure the RMSD between how ESM fold thinks that predicted structure is with the held out PDB structures. And then we can see here compared to protein, which is state of the art, we see that we're very close. Even though our model wasn't specifically trained on that, it's trained on like every task, it can still get very similar performance as protein, which we think is quite promising. And there's also this folding task, which is what AlphaFold 2 and 3 does. And this is, you can imagine, much, much harder because you're trying to go from a single sequence to a much more complicated object. Here, we do quite a bit worse than ESM fold, but we think there's like reasons for this, which is that our model has like no evolutionary information. The model is also, I think, like 100 times less parameters than ESM fold. So there's opportunities to improve upon forward folding in this case. Yeah, so like the takeaways is that we developed mostly this flow matching over discrete state spaces, which didn't exist before, but it is like taking off. Like if you think about what companies like Google, Facebook, and Microsoft are interested in, all of them are looking into flow matching over discrete state spaces. So whether you're into like biology or machine learning, I think it's a very interesting kind of new direction in machine learning that one could look into and do cool stuff in. And we're also like SOTA on co-design and competitive with protein PNN. And I think it's more of a proof of concept that I believe we're going towards this stage of trying to solve every like protein modeling task jointly, rather than having like a specific model for every single one of these arrows. As someone in biology, you don't want to have a model for each of these. That's gonna be really hard to keep up to date, and just going to make your workflow a nightmare if you have to have a model for every single one of these. Sorry, a different model for every single one of these. I think having it all in one so-called foundation model is going to be the path forward, and what I think next generation researchers should be thinking about. Yeah. So technical summary, I discussed frame diff, which is this like diffusion approach to structure generation. There's this frame flow, which then there's like flow matching over the Riemannian and SC3 space. And then this discrete flow with multiflow, where now you can do both sequence and structure flows. And so kind of like outlook of on top of this foundation model, general purpose modeling, kind of what's next. I think it's very clear that's going to be biomolecular interactions, where we can now condition like small molecules, also like RNA, DNA, nucleic acids, and AlphaFold 3 and RosettaFold all atom are the prime examples, kind of the headlining papers of this. And the next things are going to be doing design with these models. And so, I think this is a very exciting time, especially now that AlphaFold 3 is out, and also my lab, if you've heard Boltz 1, it's an open source version of this. Open source is also becoming very important, but academics are making sure these state-of-the-art models are open source. And if you're interested in doing research, this is something that seems competitive, but I think very fun and very impactful if you can work and make some progress on this topic. So here are like some references, and then 1 o'clock now, so happy to take any more questions. And here's like my contact information if you want to reach out. And then just thanks to my advisors, Regina and Tommy, who supporting me through my PhD. Yeah, cool. I think that's it. Yeah.
All right, thank you so much for the great talk. Now I think we can take some questions. And for audience on Zoom, also feel free to unmute yourself
I think so. So one thing that's important is that once you move to all atom generation, you need to know the sequence because the number of atoms in the side chain is going to depend on like the identity of the residue. And so I believe once we move to there's like all atomic interactions, cogeneration is going to become almost a necessity. I mean, there are ways to not do it, but I think it's going to be a really good way to frame the problem. And also, I think like yeah, like what we showed is you do get benefit out of doing cogeneration by like efficiency by just being a lot faster. But also, there's a lot to explore by training on like larger sequence data sets. That's something we didn't do. You could train on structured data sets, but also generate on sequence data sets, which there's a lot more data for. And so it's an open question of like how do you incorporate all the data sets together that I think if someone can do it right, should be the best performing model overall.
Further questions? So I have one relating to the discrete flow. You mentioned about using cross entropy loss being ideal, but I was wondering, like for the flow matching, for example, the original like loss was kind of proved for like L2 loss, right? So like why is cross entropy better loss?
Yeah, yeah. I guess my motivation for cross entropy was that it's the same loss that's used in language models. Um, And like doing L2 over discrete space is kind of weird. It's not the right thing. Cross entropy is the right thing. But then that said, there are other losses out there. You can do different losses based off different divergences. Um, but I think one thing is that we know it scales with large language models, that it works well there. That's kind of like my only intuition. I think it's a very active area of research. There's a lot of people at big tech companies exploring different losses with discrete flow matching. So we'll see once the research comes out of what the best loss is.
I see. But is it, I think the original like Alto loss was used because they have the like nice proof of the condition, the equivalence of gradient between conditional flow matching and full matching. I wonder these like new losses, the same kind of equivalence they will hold, or like would that break anything in the kind of the marginalization of the full matching path?
Yeah, yeah. I'm also not clear as well. Um, yeah, I think once you're working in discrete spaces, like the cross entropy is like the right right minimizer of the probabilistic divergence. And L2, I don't know of a principled way to get that from in the discrete flow. So it might be possible. I'm not saying it's impossible, but it would be nicer to have them be almost exactly the same. But I think uh, forcing them to be exactly the same might not be the right thing either.
Well, thank you so much. If there's no other questions, oh, there's one. Yeah.
What's your perspective on accounting for like additional design considerations when moving in like in vivo? Like for example, an antibody might work very well in vitro and have good binding affinity, but then when used as a drug, it can have different performance.
Yeah, I think so no one knows the relationship yet of like how to well, I mean people are working on it of how to make these in silico methods and also the in vitro methods translate better to in vivo. Um, so I think it's an open question. I think one thing that's interesting is that if something fails in vivo, could you feed that back into the model and tell it to not sample those same things or learn from that data and sample better things? And I think there's kind of a data generation and like a modeling issue there where people don't really generate data for that purpose and curate it that we can actually feed into machine learning models. So I guess like what I'm saying is like as humans, it might be hard for us to understand why something failed, but maybe if you like fine tune a generative model on the failures, you can have a better sense of why it failed and then generate better things as it learns from the like lab in the loop style. I think that's the term that's going around like having experimental data go back into your model and iteratively improve.
Thank you.
Any other questions?
Oh, yeah. I mean, I think it's coming for sure. AF3 is modeling everything in protein structures, but because I think we have structure data, we also have protein sequence data, we also have genomic sequence data. There's also a lot of this activity data as well out there. I think it's possible to combine everything together. There are papers that are combining genomic data sets with protein data sets. ESM Fold is combining protein sequence data sets with protein structure data sets. Um, so it feels like we're on the precipice of someone combining them all and maybe beating everyone at the same task. AlphaFold 3 shows you can now model all possible molecular structures. So yeah, it could be there. If you want me to take like a wild guess, in the next 5 years for sure, someone's going to build a foundation model that's going to, maybe become like a virtual cell that's like a lot of people are very interested in, and actually simulating a cell that we can learn from and actually, help bridge the gap between in vitro and in vivo. Yeah, I think it's coming. I think data consideration of generating higher quality data, we can actually fine tune it to do a specific task. That's something we also have to consider. Like, what is the actual use of the foundation models? Um, but there's a lot of foundation models being proposed, like Evo and SCGPT. For proteins, it's like ESM2 for sequences. For structures, AF3 might be an example of it. But I think I have to see for structures soon.
Cool. Yeah, I guess if there's no other questions, we can end the class today. Thank you so much, Jason, for your time.
Okay, thanks. See you, everyone.
See you. And before we leave, please remember to sign up for the attendance sheet. It's in the first row.
location in the sequence. So
Token location?
yeah, like
Promoter?
promoter, yeah. So can we do it like with fine tune, or we add some simple layer on the DNA bird too? So
Oh, there's a model called SCI, I think, SCI. You can check out.
SEI.
Yeah, SCI, it predicts the promoter profile.
Sorry, can you repeat?
SEI.
SEI,
SEI.
okay. So, like, can we still use the bird to detect the location of the promoter?
Sure, you can train your own model.
And then we kit out the DNA chisel.
You could try it. I don't know how easy to train such a model.
That's my question. I never done something like use the bird to detect the location. I'm not sure if that work. So did you try?
Why not? Like,
did you try?
Basically, just take the embeddings and predict if the next token is within the promoter region or not. Right?
So right now, what we done right now, like we add a simple layer over the DNA bird. Oh, sure.
Yeah.